I'm going to be telling you today about Rastermaps. So this is an algorithm for extracting structure from high dimensional neural data. And so the problem we have is we have these really large scale recordings of thousands of neurons. So for instance, here's an example of what kind of you want to plot your large scale neural recording. You have 50,000 neurons recorded. In this case, this is in primary visual cortex of a mouse, and you have this time axis here. And now the question is, how do you make a visualization of this data so that you can try to find patterns inside this data? And so we created an algorithm basically, which puts neighbors kind of in the original space, this high dimensional space that you have many time points in, that puts these neighbors in this original space close to each other in this low dimensional, one-dimensional embedding space. So these neurons are put next to each other along this axis to start to reveal patterns of neural activity. And at the top, you can see the running speed of the mouse, but you can see there's more patterns than just neurons being driven by running. There's lots of different patterns here. And this is a mouse in total darkness, so there's no actual visual input to visual cortex right now. So there's lots of these different patterns of activity that can be revealed if you do this kind of resorting. And so I'm going to be telling you about Rastermap today, which is a one-dimensional embedding algorithm. And so you could imagine for people who've used t-SNE before, you can run t-SNE in one dimension too. So it's kind of doing the same sort of thing as t-SNE, but we have some tricks to find dynamics in the patterns that t-SNE doesn't have. And we also outperform t-SNE and UMAP on these kind of standard metrics that people use to benchmark embedding algorithms. All right, so we want to preserve the original structure of the data. And so we actually, we enforce kind of a property of the data that we found in the past, that the high dimensional properties of the data. The data that we record isn't just high dimensional in an arbitrary way, it has a special structure. So I'm going to briefly show you what that structure is and then how we enforce it to create this embedding algorithm. Okay, so we asked this question, kind of what is the structure of neural activity? And so we have, we're looking at the visual stream. So we have images that come into the retina and they get transmitted to the thalamus. And then these signals get transmitted to visual cortex. And there's around a 50 times expansion of the number of neurons in visual cortex compared to thalamus. And this is true in mice and in primates. And so there's this question that what is the structure of the activity in visual cortex and what does this expansion mean? So one hypothesis is that this expansion means that every neuron in thalamus drives 50 neurons in visual cortex in the same way, and they all represent the same kind of thing, and you have this redundant low dimensional circuit. So that's one hypothesis, is that visual cortex is reducing noise by having many neurons representing the similar things. Another hypothesis is that these visual cortical neurons combine these thalamic signals in diverse ways and in unique ways, creating a really large feature space, and in other words, a high dimensional representation of the natural world. And this hypothesis is appealing because if you transform your data into a high dimensional space, then you can start to make problems which are nonlinear in the low dimensional pixel space more linearly separable, for instance. So like, for instance, a deep neural network takes images as input and transforms them into a high dimensional space, which makes it easier to make linear classifications like this is a tiger versus, and this is not a dog or something that you should be scared of the tiger and be able to quickly react to that. And so it makes kind of these types of image recognition problems easier. And so we have these two hypotheses, and we wanted to ask which one is true in particular in mouse visual cortex is where we asked this question. And so, in fact, even though a high dimensional code might be appealing, in many cases, many studies have hypothesized that neural codes are low dimensional in visual cortex and in other brain areas. So for instance, an example of a low dimensional code would be one where you have a one-dimensional stimulus, and here are tuning curves that are very broad to this one-dimensional stimulus. So if you have these broad tuning curves along a single axis, you're going to basically, you're going to smoothly represent this one-dimensional stimulus, but you're not going to represent perhaps the fine features of that stimulus. And so a low-dimensional code like this will have only two significant eigenvalues. And it will, if you take a random projection of this neural activity of this simulated neural activity with these large tuning curves, and you move in this one-dimensional space, you smoothly kind of move along this circle. So this code is robust to noise, but it maybe is not representing very fine features of this one-dimensional stimulus space. On the other hand, so other studies have posited that neural activity is high dimensional. And so they've taken, you can imagine a one-dimensional stimulus now, and you create a population code where every neuron represents a different position along this one-dimensional stimulus. Like if this is orientations of a grading, one represents 1 degree, one represents a 2 degree grading, 3 degree, all the way up to 360. And so this would create an eigenspectrum that's flat, where the variance is equal for every single eigen direction, because every single neuron is its own direction, because there's no correlations in this space. And so this is a type of efficient code where the input is entirely decorrelated in the population activity. And so this might be advantageous because you can represent finer features of the stimulus space, but at the same time, you're losing the smoothness property. So if I move only a little bit in the stimulus space, you basically completely change your representation to another neuron. There's no smoothness here. So if you try to move around in the stimulus space, in this random projection, for instance, you have this spiky ball that you're moving from. Your representation changes completely because a completely new neuron is firing along this axis. Okay, and so many studies in the past have suggested that neural activity is low dimensional, and so that maybe we don't even need a high dimensional embedding algorithm to understand our data. But I'm going to show that that's not the case, that neural activity is high dimensional with a special type of structure. So these low dimensional studies, had that a positive that neural activity is low dimensional. So there's several of these studies. There's a nice review article by Gao and Ganguly from 2015, which summarizes the dimensionality of various neural recordings. And many of these recordings have a dimensionality less than 10. But this review paper and also another paper from Byron Yu's group state that the dimensionality of the data is limited by the number of neurons and the number of stimuli that you show. So you can't have more dimensions than the number of kind of task parameters you have. And so many of these studies, for instance, will only record 10s to 100 neurons, and they'll only have maybe 8 different stimulus conditions, like the monkey reaches in 8 different directions. And so that automatically limits the number of dimensions that you can find in your neural recordings. And so we wanted to avoid being constricted by these constraints of the number of neurons and the number of stimuli to try to, as best as possible, estimate the dimensionality and the structure of the neural activity. And so basically we did really large recording, and we also showed many thousands of visual stimuli in a single session to these mice. And so now we're not limited by the experimental setup in order to find these different dimensions. And so how do we record many thousands of neurons? So we use 2-photon calcium imaging, and when we use a microscope, which can record at many depths very quickly. And so this is one plane, and we record, for instance, 12 planes at a rate of 3 hertz. And in each of these planes, we'll capture at least around 1,000 cells. And so we can get pretty reliably at least 20,000 neurons per recording. These recordings were done in my old lab in London, so these are more around 10,000 neurons. But generally speaking, we're getting many more neurons now, even with newer microscopes. And so here's a video of what the neural activity looks like. So each of these squares is a plane, and these neurons light up when they fire. And when the visual stimuli come in to the mouse's eye here, so they're being shown in a screen in front of the mouse, the neural activity increases greatly. So there's neural activity when the mouse isn't seeing anything on the screen. So you'll see here in a period when there's no visual input, there's still activity. But then when the visual stimuli come in, you can see lots of these neurons being driven by these various visual stimuli. And so now we can take this neural activity and we basically have an algorithm, SUI2P, which finds the little disks here that are flashing and segments them from each other, and then we can basically sum the intensity of those pixels at every single time point, and that gives us the activity of that neuron at every given time point. And so we're doing flashed images, and so we flash 2,800 different images, and we have each image on for around 750 milliseconds. So that will be around 2 or 3 samples of our imaging time, because we're imaging at a rate of 3 hertz. So we average the activity in about 2 time samples for each neuron for each stimulus. And so that gives us a matrix that's maybe, if we have 10,000 neurons, it's 10,000 neurons by, if we show 2,800 images, basically. So we have this matrix now we're going to work with, and we're going to try to find its eigenvalue spectrum. And I'm not going to go into the details of how we find an eigenvalue spectrum despite the kind of trial-to-trial variability that neurons have. I recommend that you check out our paper. But we basically, we don't show every image once, we show it twice. And so that helps us to try to find the stimulus-specific eigenvectors, not, and we don't want to find noise directions, basically. So yeah, you can check out this paper for more details on how we compute those. But basically, let me get to the punchline. So we asked this question, is neural activity low dimensional or high dimensional? We want to know what the structure is so that we can design better algorithms, basically, to find, to understand its structure. And so we computed the eigenvalue spectrum for the neural activity that I showed you on the previous slide. And we found that it was neither low dimensional in that it had only a few significant eigenvalues, and it was not high dimensional in an efficient coding sense where the eigenspectrum was flat. Instead, we were surprised that we found something that looked like a power law decay, where basically the variance is 1 over n to the alpha, where alpha is close to 1. And so this means that there's these eigenvalues at the top which have more variance, which kind of can allow for potentially the space to smoothly vary across stimulus dimensions, unlike this efficient code, which doesn't have these significant top eigenvalues. But there's still many thousands, over 1,000 significant eigenvalues in the tail, which can encode finer features in the stimulus space. So we think that this suggests that the neural code is balancing 2 things. So capacity, you want to maximize as much information as possible that you can encode about the sensory world. So you want to be as high dimensional as possible. Like what I was saying before, for instance, to do various visual tasks, it can be helpful. But you also at the same time want to have smooth representations. So you want similar stimuli, for instance, to be represented similarly if you're trying to do things like generalization, or even just being robust to noise within the system, or noisy stimuli from the world. And so we're going to actually enforce this power law decay when we're trying to optimize our algorithm for embedding these neurons. And so that's kind of why I went through this, just so that it's not an arbitrary high dimensional structure we're trying to embed. There's actually some structure that we can be inspired by. And it's not just because we saw it in this data, we also saw it across various data. So in stimulus, this is the stimulus-driven eigenvalue spectrum. If we look at spontaneous activity, so this is when the mouse is in the dark and there's no stimuli coming into the mouse. Basically, we're not showing stimuli on the screen, and we're looking at visual cortex activity. We also see a power law decay of the neural activity. If you look at zebrafish whole brain activity, you see a power law decay. Natural images are kind of the classical case of having a power law decay of their eigenvalues. And you can see this kind of nice property of natural images, this self-similarity property here in this nice zoom in here. And then the English language, for instance, if you take the correlation of words across sentences, it also has a power law decay. And finally, social networks have also been explored in this way, and they also have this power law decay. And so we think this is a reasonable kind of constraint to put in our algorithm because we think it exists across many different data types. Okay, so how do we create this algorithm that puts these neurons close to each other? And so I'm going to go through kind of the 4 steps of creating this Rastermap plot, and then I'll go through how we apply it. Okay, so the first step is we reduce the data to a manageable size. And so we've got, in our new latest recordings, we're able to record 50,000 neurons at 3 Hertz. And so in these really large scale recordings, we first want to reduce the data to a size that we can deal with. And so to do that, we do Kmeans clustering, and we can extract around 100 to 200 clusters from these neurons. And the reason we choose K-means clustering rather than another clustering algorithm is because it gives us kind of a covering of the space. And so we expect each of the cluster centers to hopefully be close to at least some subset of neurons in the data. But you could imagine doing other initial pre-processing steps to get a subset of clusters from your data, which could be running NMF or other things like that. You could run it in a variety of ways. But basically, this is kind of the standard way that we run it. We do K-means clustering. We get 100 to 200 clusters. And now what do we do with those clusters? So basically, we're trying to create this sorting where neurons, or in this case, clusters, are put next to each other if they're correlated to each other. And so we're going to look at our cluster correlations, which makes a matrix like this. So each of these clusters has neurons. Basically, every neuron is assigned to a cluster. And you can imagine, so then we can take the average of the neurons in each cluster. That gives us a time trace. And so we have a time trace for every cluster. We can correlate these time traces, and that gives us this cluster correlation matrix. And so we want to rearrange these clusters by similarity. You can imagine one thing is you try to put every cluster closest to its most correlated cluster. And that's sort of like a traveling salesman problem, where we're trying to optimize just the off diagonal of this matrix so that the most correlated cluster is closest to the other, to its most correlated neighbor. But we don't just enforce we don't enforce that penalty alone. We think it's important to enforce this kind of power law decay of the eigenvalues. So a matrix that has a power law decay of eigenvalues has kind of this heavy tailed decay along the off diagonal. And so this is so that we don't want to just explain local structure. We also want to explain the global structure of the data. So we're not just going to put clusters close to each other that are most correlated to each other. We're also going to try to put groups of clusters next to each other that are globally correlated to each other. And so we're basically trying to optimize this matrix to look as much as possible like this matrix. So we're basically, on every iteration of the optimization, we're doing a dot product between this matrix and this matrix. And we basically, we take, for instance, we have a sorting, which is, we have 100 nodes, for instance, in the sorting, 100 clusters. And at every step in the optimization, this iterative optimization, we ask if a given segment of two nodes, if we move this given segment to this position, basically we try every single position and we ask, do any of these positions increase this score, increase this dot product between this matrix and this matrix? And if it does, then we move those nodes there. And we continue this iterative optimization, trying many pairs of nodes. And we only try subsets of nodes that are of length at most 3 to try to make this optimization faster. And so this is similar to the types of optimizations people use to solve traveling salesman's problems, but instead of just trying to optimize a path, we're optimizing a dot product of these two matrices. And so, yeah, there's a lot of talking, but this is what the end result actually looks like. So we have a cluster by cluster matrix, which before looked kind of random, and now is basically has a lot more weight along the diagonal. And so there's lots of clusters that have correlated activity to each other that are grouped together along the diagonal now. And so we've not just put clusters, like I was saying, that were most correlated to each other next to each other. We've also tried to preserve some of this global structure across tens of clusters. And so then the next step is, so so somewhat unfortunately, so this is for people who have run sorts of these kinds of NP hard optimization problems. They don't necessarily scale well. So we can't really run this optimization for more than 200 clusters. So we're limited to these 200 clusters, but we can do a trick to try to get more kind of clusters in a sense in the next step, which is we upsample between these cluster centers. So we have maybe 100 to 200 cluster centers, and we take, we do linear interpolation between the cluster centers with a factor of 10, for instance, and that will give us 10 times more positions to try to assign neurons to. And so in this case, we ran Rastermap with 150 clusters and an upsampling factor of 10. And so every neuron was assigned to its position, which it was most correlated to, of these 1,500 positions. And this is a plot, a histogram of how many neurons were assigned to each position. And you can see it's relatively consistent. There's not huge spikes everywhere, so there's neurons that are throughout kind of this that are put throughout this axis. So now the final step is I have a position for every neuron. There's 1,500 of these different positions. I'm going to put them into their positions here along this axis. And then something we do, this is kind of optional, but it helps us to visualize the data better, is we create these super neurons. So these neurons are now sorted along this axis, and we can bin groups of 50 neurons, for instance, along this axis, to give us kind of these super neurons, which are like these less noisy versions of our original neurons. And so this is advantageous. So a lot of interesting things that a mouse is doing happen on a single trial level. Like the mouse's behavior changes moment by moment, like our behavior changes all the time. And so being able to average over neurons can be advantageous to get rid of some of this noise, because we don't necessarily want to average over trials because the behavior can vary from trial to trial. So these sorts of super neurons might be easier to analyze trial by trial variability, for instance, rather than other using the neurons directly. Okay. So that's how the algorithm works. And so I think this is useful, particularly for people who don't necessarily build embedding algorithms all day, is how do you benchmark these types of algorithms, and what are you looking for in your embedding algorithm? And for people who are on Twitter, there was recent uproar about how t-SNE is not good. And it really depends on what you're trying to get out of these algorithms. And so in our case, I'll tell you the kind of benchmark we think is important for our data, and what people often use in the nonlinear embedding field to benchmark their algorithms. And it may not be what you're looking for in an algorithm, but it can be helpful in our case, we think, for visualizing neural activity. Okay, so basically what we think is important is that an embedding preserves local neighbors. So what this means is, so here in this toy example, we have 3-dimensional data. So you can imagine I have 3 different visual stimuli I show, and this is the response of each neuron to these different visual stimuli. So I have many of these different neurons. So like, for instance, these green neurons all respond pretty similarly to these 3 stimuli. The blue neurons respond similarly, and so on. And so basically, if I make an embedding that preserves local neighbors, I expect that all the neurons that respond similarly are put next to each other. So all the green neurons which respond similarly should be put next to each other in the embedding. All the blue neurons should be put next to each other, all the red, and all the yellow. And so we expect we expect that local neighbors should be preserved, particularly if we want this property, like I was saying, where we average over groups of neurons that are next to each other in the embedding to try to reduce noise. This score is very important for keeping it to be able to do that kind of trick, for instance. And so the way you benchmark how well you're doing in preserving local neighbors is you ask how many of my original neighbors in my high dimensional space are still my neighbors in my low dimensional space. And so I can take, for instance, my top 5 neighbors in my original space and check, are they still my neighbors in my low dimensional space? And you can ask what fraction are still your neighbors in your low dimensional space. And so we did that for Rastermap, for 1-dimensional t-SNE, for 1-dimensional UMAP, and the first dimension of PCA. And what we found was for the top 10 neighbors, Rastermap preserved more of these neighbors than t-SNE, UMAP, and PCA. Basically, it did a better job of preserving these local neighbors of the neural activity. And note, we ran t-SNE with a variety of perplexities. And this was kind of, so I could, in theory, increase the local score for t-SNE, but I at the expense of reducing the global score. And so I'll explain that now too. So you might also not just care about increasing this local score, you might also care about preserving the global neighbors. So for instance, if I zoom out, am I preserving the global structure, which might help me to see these patterns and how they're related to each other in my embedding. And so to do that, you literally just start taking more and more neighbors. So instead of taking 10 neighbors, for instance, you might take 100 neighbors, and you see if that structure is preserved. And you can see, for instance, in this toy embedding that I created, the local neighbors are preserved, but the global neighbors aren't. So for instance, these green points should probably be closer to these blue points, and they're not. And so something that would preserve global distances better would, for instance, be something that maybe looks more like this, where now the green points are next to the blue points and the yellow points. And there's always this kind of trade-off between there's like a tension between these things, because the more you preserve the global structure, the more you might be losing kind of the fine local structures in the data. And so that's a point to make here. Like, so as we zoom out, if we take 100 nearest neighbors, you can see our score is still high for Rastermap, t-SNE, and UMAP. And then if you zoom out even more and take the top 1,000 neighbors, you can see that an algorithm like PCA, for instance, does a pretty good job on this score, but you can see it preserves a global structure at the expense of not doing well with the local score. So you can kind of have trade-offs between these different things. So it kind of depends on what you're looking for. But basically, Rastermap does at least as well or better than these other algorithms on the intermediate and global score. So we think it's the best in terms of preserving the actual structure, the high dimensional structure of the data, of neural data. Okay, so now let's show some examples of Rastermap. So this is actually in the paper, so I'm gonna skip over this. Basically, we took different principal components, and you can sort not just the neuron space, you can also sort the image space or stimulus space with an algorithm like Rastermap, or even t-SNE if you're not using Rastermap, and you can see different structures of in this case, we're looking at the different principal components and how many neurons respond to different sets of images in this space. And for instance, you can see the top PCs generally have more of this global encoding of the image space, whereas if we look at higher PCs, there's smaller subsets of neurons responding to smaller subsets of images. And then we also have applied, so my student Ada Casillita is working on this data. Basically, we can look at how neural activity is driven by the behaviors of the mouse. So we can, for instance, take the mouse's face, and so this is what the mouse is doing during this recording, and we can take some principal components of the mouse's facial movements and replace every neuron in this plot. Basically, every prediction of each kind of, you could use neurons or super neurons and replace each row here with the prediction from the behavior. And so having an embedding algorithm like this with this kind of sorting allows you to see, you can see kind of what types of dynamics is your prediction capturing and what is it not capturing. What sorts of what sorts of things is it missing across neurons? So you can look at residuals, that sort of thing. So another example of where you can apply Rastermap is to behavioral data where a mouse is doing a task. So here's an example of a nice task setup that Lin Zong has set up. He's a postdoc in Marius Pagliaru's lab. And so he has set up 2 linear corridors. So one, they're visual corridors. So the mouse is running in a virtual reality. And in one corridor, the mouse is rewarded. So in this red corridor, the mouse gets rewards at these green times. And in the blue corridor, the mouse is not rewarded. And so whenever the mouse gets a reward signified by this green line here, the mouse actually stops running, and you can see it kind of, and then it picks up running again. You can kind of see, it looks like there's starting to be maybe sequences in this data, like visually driven sequences of neural activity in primary visual cortex. And we couldn't see it so well. We have a trick basically to incorporate dynamics information into Rastermap using time lag correlations rather than instantaneous correlation. So we actually transform our correlation matrix So instead of taking the instantaneous correlation matrix, we take the maximum correlation across time lags, maybe like, for instance, in this case, I think 4 or 5 time lags. And if we do that and we use that matrix instead of our instantaneous correlation matrix, we can actually capture these sequences. And so here's an example of now when I resort this matrix, you can see these sequences clearly, that as the mouse runs through this corridor, these neurons are driven in a sequence basically across the visual cortex. And so this is the sequence for this corridor. You can see these stops here is when the visual flow stops because the mouse stops running. And so the mouse stops, in this case, it's licking for the reward and then starts running again. And in this case, again, the mouse stops, it licks, and then starts running again. And so you can see this across these different corridors. In the blue corridors that are unrewarded, you can see the mouse is just running nice and fast straight through because it knows there's no, it learns that there's no reward in that corridor basically. And it doesn't lick often in that corridor. You can see that this green is the licking. Okay, so one of the nice things about Rastermap being a one-dimensional embedding is that you can color neurons along this embedding based on brain area. And so we can kind of go through the embedding and see where are neurons in different across the brain. And so some of these neurons can be really localized to maybe, so we're looking at visual cortex, maybe to higher, that's a higher order visual area, that's more primary visual cortex, higher order visual area. In the sequence, there's more neurons in these in the back part of the brain, but there's also some neurons in higher order visual areas being driven too. Okay, and so you can also run this on zebrafish data. So here's an example. This is neurons by time, but now with zebrafish, their larval zebrafish in particular are transparent, so you can amazingly do these really hundreds of thousands of neurons simultaneously capturing the whole brain of the zebrafish while it's doing various things. So in this case, in these recordings, they showed a variety of different stimuli, some of which the fish started swimming to. So down here, these are, for instance, swimming signals. And so you can see a variety of patterns that Rastermap picks up. And again, we can because we have a one-dimensional embedding, we can color neurons in the zebrafish brain according to their positions. And so there's some clusters of neurons that appear to be in different in specific brain areas, and some clusters, for instance, that seem to be throughout the zebrafish brain. And then we can also, you don't need to apply it just to single cell data. You can also apply it to data like widefield imaging data, which is basically you have voxels instead of single cells. So it's kind of like, you can think of it like fMRI data for people who are familiar with that. So you kind of, although you have fast temporal precision, more fast temporal precision with this data. But basically, here's an example of a widefield imaging data set from Ann Churchland's lab where they're recording while the mouse is doing an auditory discrimination task. And they record also, they also have trials that are always rewarded where they show visual stimuli too. And what we found when we sorted this data is that voxels that are close to each other in the brain, maybe somewhat unsurprisingly, are also put close to each other in the Rastermap. But this gives you a way potentially to parcelate different brain areas that in a kind of an agnostic way, where you're paying attention to the activity, not necessarily to what the atlas of brain areas tells you. You care about putting neurons voxels close to each other that are correlated to each other, not necessarily where exactly the anatomical boundaries are. Okay, and then this is the last thing. So you don't just have to run this on neurons in the brain, you can also run this on artificial brains. And so here's an example of an agent that's been trained to play Space Invaders. And you can see these so this is neurons by time. And so I've basically concatenated, so this network consists of a convolutional neural network, which processes the game images, and also a value network, which has a few feed forward layers. And so I've concatenated those and sorted them with Rastermap. So that's why you can see maybe, for instance, this is probably the neurons from the value network. And then you have neurons from the convolutional neural network, which have these sequences, which are visually driven, just like the sequences that we saw in the mouse, although the mouse isn't playing Atari. But you can see basically this agent playing the Atari game. And basically what happens at this breakpoint where things change is exactly when the agent clears the screen here. And it's about to succeed. And then it clears the screen and it continues onwards. And then you see some of these similar patterns repeated again as it goes screen by screen. We also tried it on, so there's lots of these kind of pre-trained networks on different Atari games. Here's one on the Sequest game where the agent has to swim around and shoot fish and pick up these little divers. It also runs out of oxygen. This agent is not very smart and doesn't realize it needs to go to the surface to get oxygen, so it just dies every time. But that again, like these changes in what happens every time it revives itself and these screens change again correspond to these large changes you can see in the raster map. So it's picking up kind of these sorts of changes here. And yeah, so that's it. Hopefully you have a sense of how Rastermap works. And I've shown you that Rastermap is able to capture this local structure better than t-SNE and UMAP. I showed you an extension of this algorithm for enabling sequence finding, like those sequences, those visually driven sequences as a mouse runs through a virtual corridor, also sequences in the Atari game. We can also we can extend the algorithm for fine scale structure discovery in high dimensional data, but I didn't show that today. We also have a GUI to explore the data, which Attica built, and I can play this in the background where basically you can load in your data and run the algorithm inside this graphical interface, and you can also load in your own behavioral variables to see how they vary with these various neural patterns. And so I'd like to thank Marius Pakhtsaryou, my close collaborator, and also Lin Zong, who did those really nice behavioral experiments that I showed you, and also my student Attica Saida. And thank you for inviting me to this seminar series, and thanks everyone for listening.